afternoon. Well, welcome to today's lecture. This is the Solar Distinguished Lectureship Series. And it is indeed a privilege and a great opportunity for us to have with us Professor Robert Langer from MIT. Before I introduce him to you, I would like to say a couple of things about the Solar Lectureship Series, which, as you know, is the most distinguished, the most respected such lectureship series that we have on the UC Davis campus. And this is due to the generosity of Professor Tracy Storer and Dr. Ruth Storer, who endowed this lectureship series in the 60s for the sole purpose of bringing to this campus some of the most eminent people in the biological sciences, in biomedical sciences, in biomedical engineering, and it is indeed a great opportunity that the Dean of the College of Biological Sciences gave us the opportunity to have a biomedical engineer be the story lecturer this time. Professor Langer, let me tell you a couple of things about Professor Langer, and of course, it's not going to be an exaggeration if I say that if I were to read about what he has done or what he has accomplished, I would take the next 2 hours just talking about himself. And I'm not going to do that, but I'm just going to tell you that we are in the presence of a living legend. Professor Langer is, of course, a man who is considered by many people, most people, probably the most important modern engineer in the world. Let me give you a couple of examples of what he's done. He has published in excess of 1,000 full-size publications, peer-reviewed publications. He has authored or co-authored close to 800 patents. And 220 companies have taken over some of his patents, licensed the rights to some of his patents to commercialize products that are routinely used throughout the world. So he has received over 180 major awards, including the 2008 United States National Medal of Science. He received the Charles Stark Draper Prize, which is considered to be the equivalent of the Nobel Prize for engineers. As you know, there's not a Nobel Prize in engineering. Well, this is it, the Draper Prize. He won the 2008 Millennium Prize, which is the world's largest technology prize. He's the only engineer to receive the Gardner Foundation International Award, and I would be remiss if I did not indicate that 72 of the previous recipients of that award are people who received subsequently the Nobel Prize. The list goes on and on and on and on. In '98, he received the Levelson MIT Prize, the world's largest prize for invention, for being, quote unquote, one of history's most prolific inventors in medicine. That was in '98. So you can calculate how much more he has invented since that time. In 1989, he was elected to the Institute of Medicine of National Academies. And then 2 or 3 years later, he was elected to both the National Academy of Engineering and to the National Academy of Sciences. So he's one of very few people who are members of all three academies. And he is the youngest ever, at the age of 43, who became a member of all three of the academies. So he received his bachelor's degree from Cornell in 1970 and his doctorate from MIT in 1974, both in chemical engineering. So he's considered to be the father of tissue engineering. He's considered to be the father of controlled release. He's considered to be one of the fathers of modern bioengineering. So it is an absolute pleasure and it is an honor to introduce Professor Langer. Please join me in welcoming.
Yes, I got it. Yes, I got it. Oh, I like that one. Okay. Remind me not to keep it, though. I like green ones. Thank you. Thank you. It's really a pleasure to be able to speak with you today. What I'd like to do is go over several topics on our research and talk about some of what we've done. But before I do that, in these three areas, I thought I probably should tell you a little bit about myself and how I got into this field, because it might seem strange. How does an engineer get involved in medicine in some cases? So just to turn the clock back, so for me, I got my doctoral degree at MIT in 1974. And for those of you that are old enough to remember, probably not that many as I look at the audience, but there was what we heard was an energy crisis then. And the consequence of that, just like you heard about a year or two ago, and not only did the prices of gas keep going up like it did a couple years ago, but it was actually worse. If you lived in Boston, where I live, it turned out that you'd have to wait in line with your car for about 2 hours to get the gas. But if you were a chemical engineer, what it also meant is that you got lots of job offers. There were all these oil companies and gas companies like Exxon, and they just had so many job openings. So I actually got 4 offers from Exxon alone. And it wasn't like I had spectacular grades or anything, but they just had so many job offers. One of them actually made quite an impression on me. And I should point out that pretty much every one of my classmates in 1974, that's what they did. They went to oil companies. One of them, I remember, though, one job interview I had was at Exxon in Baton Rouge, Louisiana. And when I was there, and I did the interview, one of the engineers said to me, he said, if you could just increase the yield of this one petrochemical by 0.01%, he said, that would be wonderful. He said, that would be worth billions of dollars. And I remember thinking to myself as I flew back to Boston that night that I really don't want to do that. So I started thinking more about what I would do, even though everybody else in my class was going into the Exxons of the world. And one of the things I did when I was at MIT is I spent a lot of time helping start a school for poor kids. And I did a lot of work developing chemistry curricula and math curricula. So one day I saw an ad for an assistant professor at City College of New York to develop chemistry curricula. And I thought, this is great. This is what I want to do. So I wrote them a letter. But they didn't write me back. But I liked the idea, so I wrote to about 40 other universities to see if I could be assistant professor in chemistry education. And I think only about 2 wrote me back, and they were very thin letters. I didn't get any offers there. So then I started thinking, but I always wanted to use my education to help people. Started thinking, what other ways could I do it? And one of the areas that I thought about was medicine. So I wrote to a lot of medical schools and hospitals. They didn't write me back either. And then one day, one of the postdocs in my laboratory said to me, he said, Bob, he said, there's a surgeon in Boston named Judah Folkman. And he said, sometimes he hires unusual people. He actually thought very highly of Dr. Folkman. I won't say what he thought of me. So I wrote to Dr. Folkman, and he was kind enough to offer me a position. And I thought I'd actually start this talk with the first part of the talk and just go over the problem that Dr. Folkman wanted me to solve. And the picture I'm going to show you is from 39 years ago in the New York Times. And it was Dr. Folkman's vision of how tumors grew. Still pretty accurate today. And what he said is that somehow a tumor cell became abnormal, started growing as a 3-dimensional mass. But it would not be able to get beyond a very tiny size of, say, about 1 millimeter because of a nutritional problem. Cells in the center couldn't get nutrition or couldn't get rid of waste. Cells on the outside could, right, because they're exposed to the environment. And they couldn't get bigger than that. But he said what the tumor does, in contrast to other tissues, is it actually sends out a chemical signal, which he called TAF, tumor angiogenesis factor. And see, the blood vessels that we have, they're normally quiescent. They don't change. They don't divide. But in the presence of a tumor, they do. And what he said is that normally the capillaries would be stagnant, but in the presence of the tumor producing the signal, they start to multiply very rapidly. And they feed this little tumor mass. And because they do, they vascularize it. That solves the nutrition problem for the tumor. Now, through the blood vessels, they can get the nutrients. They can get rid of waste. And the tumor grows and becomes bigger and bigger. And ultimately, it could spread through those blood vessels and do what's called metastasize and kill. Dr. Folkman said if you could stop the blood vessels, maybe you could stop the tumor. And my project, actually, as a postdoc, was to figure out the first substance that could stop the blood vessels from growing. And it was a big challenge. What could stop blood vessels from growing? So we thought there's a tissue in our bodies called cartilage. It's in your nose or your knee. Cartilage has no blood vessels. And so we started getting cartilage from baby rabbits. But you can't get that much cartilage from the rabbit. So, if you're an engineer, you think about scale up. So I went to cows. Cows had much more cartilage. And we got a cow's bone from a slaughterhouse in the Boston area. But I still couldn't get enough. So I asked them, I said, well, where do your bones go when you're done? And they said, well, they send all of them to this meat packing place in South Boston. So I made an arrangement with this meat packing place in South Boston to get all their bones. And let me just show you one of them. Now, it doesn't come like this. This is the bone. This is actually the cartilage. When you get the cartilage, actually, there's a lot of meat on it, the residual meat. And so what I had to do was I spent probably 40 hours every week as a postdoc scraping meat off these bones to make it look like that. But this is pretty typical. It makes it look like that, and it's quite clean. And then I would take this, and I would put it through various purification procedures, and I'd have all these different things that I wanted to test to see if they could stop blood vessels from growing. Now that gets to the second problem. So one was, could we find a substance that could stop the blood vessels from growing? Maybe it's one of the things we isolated. But how do you test it? How do you test whether blood vessels are growing or not? So what we thought about was maybe we could develop an assay out of the eye of a rabbit, which normally doesn't have blood vessels, but if you put a tumor in the eye, if you put a tumor like the V2 carcinoma, what will happen is that blood vessels will grow from the edge of this. Over about 6 to 8 weeks, they'll grow over the edge of this to the tumor. They'll actually vascularize. I'll show you a picture a little later. But so we thought you could take an ophthalmic microscope and look and see how fast the blood vessels were growing. But to see if we had an inhibitor, and that's what we were looking for, what we needed was a controlled release polymer that could take any large molecule we'd isolated, put it in, and release it. So what we needed then was a polymer that was inert in the eye and that could deliver any large molecule. And of course, all the molecules in cartilage were pretty large. Now, when we started this, it was pretty basic work, interest. But as the 70s emerged, all kinds of biotech drugs, so biotechnology drugs, would also emerge. And these drugs face very serious delivery problems. If you tried to swallow a large molecule or put it in a patch, they're just too big. If you inject them, they've got short lifetimes. So really, from a practical standpoint, if you want to deliver any large molecule in active form over a chronic situation, you'd have to have a way to deliver it in an unaltered form and yet protect it from harm. But when we started this work, people didn't think you could do that. If you look in the literature in the 70s, you see quotes like this: the agent to be released is a small molecule with a molecular weight no larger than a few hundred. One would not expect that macromolecules, for example proteins, could be released because of their extremely small permeation rates through polymers. Actually, the only advantage that Dr. Folkman and I had when we started doing this work is we just didn't know that. And so I spent several years working in the laboratory, experimenting with all different types of techniques to see if I could solve this problem. And I actually found over 200 different ways to get this to not work. But eventually, I was able to make little microspheres like this. Here's one cut in half. In fact, today, we're making a lot of nanospheres for different purposes. But then you could take these and put any type of molecule in it and deliver it for over 100 days. And later on, we even were able to get constant release. Now, in 1976, I got asked for the first time in my life to give a lecture at a scientific meeting. It was in Midland, Michigan. And I was, up until that time, I'd actually only given one major talk in my life, which was in 8th grade. And that 8th grade talk didn't go very well. In 8th grade, I had to give a 1.5-minute speech. And so what I did is I wrote it out. And 2 nights before, I stood in front of my parents' mirror, and I gave it over and over again. The night before, 4 hours, I stood in front of my parents' mirror. I gave it over and over again. So finally the day came in my 8th grade class and I had to give the speech, and I unfortunately didn't bring up that sheet of paper that I'd written on. So I started out giving it, reciting it, and I was doing okay for the first minute and 2 seconds. But then I could not remember the next word, and I just froze. And I stood up there for another minute saying absolutely nothing until my 8th grade teacher finally told me to sit down and gave me a not particularly good grade. I think it was an F. And so ever after that, I was very worried about public speaking. But you know, if you're going into academics, you do public speaking, you can't avoid it. So now, many years later, in 1976, I had to give this talk. And it was a very distinguished audience of polymer scientists. And so I practiced it for 2 weeks before this talk. And finally, I got up and I gave the talk. And of course, that's 34 years ago. So I was a lot younger. You know, I had more hair. It was darker. And I was giving this talk to this very distinguished polymer audience. And actually, this time, I thought I did much better. After 20 minutes, I didn't forget too much what I was going to say. I didn't stammer too much. So I thought when I was done with this talk that these older scientists, being nice people, would want to encourage me, this young guy. And when I got done and I stepped off the podium, a bunch of them came up to me and they said, well, we don't believe anything you just said. It's actually my introduction to how scientists sometimes treat other scientists. I'll come back to this point later. So it wasn't for another 3 years later till people started repeating what we did. And then the question shifted to how could this happen? How did these large molecules get out? Well, what we did is, since I'd worked in a hospital, you know, they have all these instruments like cryomicrotomes and that they use in pathology. And so I started, instead of cutting tissues, which is what people normally did, I cut polymers. And Rajan Bawa, one of my students, did a lot of this. And this is a 5-micron thin section of one of the polymers. And if you had a molecule 300 molecular weight or greater, it can't diffuse from one side of this to the other. How do the molecules get out? Well, if you put a drug in, what you see, this is before any release has taken place, another 5-micron thin section, you see a phase separation. Polymer here, drug here. And if you release this, say, for a year, and then you cut a thin section, it really looks very different. Left behind where the drug was are these pores. And these pores are large enough so that molecules, even millions of molecular weight, can get through. But there are many tight constrictions between the pores, and the pores are also incredibly winding and tortuous. So it takes a very, very long time for the molecules to get through. One way I sometimes explain this to people, living in Massachusetts, is I sometimes explain this to people, it's kind of like driving a car through Boston. And if anybody's been there, Boston has what we call in engineering a very high tortuosity. Like Davis has a low tortuosity, but Boston, you could get lost very easily. But by controlling this pore structure, we could get release anywhere from days to years, or any time in between. So since we could do this, now we were in a position to address this angiogenesis problem. Tumors here, polymers here, and what we found, I apologize if anybody doesn't like the sight of blood, but let me just show you the next slide where we're going to look at some eyes. And in the control, without the cartilage-derived inhibitor, we see about 5 weeks after the start of the experiment, the sheet of blood vessels growing from the edge of the cornea over the polymer towards the tumor. But look what happens when you put the inhibitor in. We call it a cartilage-derived inhibitor. The blood vessels are lower. They avoid the tumor. In fact, if you looked at this just 2 weeks after this, you would see the tumor out of the orbit of the eye, and it would ultimately kill the animal. This never would. So this provided a basis then for isolating the first angiogenesis inhibitor. We published this in Science in 1976. But medicine, unfortunately, doesn't move quickly, and you need an enormous amount of champions and efforts to make this happen. It took another 28 years from the paper that we wrote in Science with Dr. Folkman, before the first angiogenesis inhibitor got approved. And this was through the efforts of many companies and other individuals working very hard. But now what's happened in the last 6 years is quite remarkable. Angiogenesis inhibitors have become one of the 4 main therapies for cancer. And you see approvals starting in 2004 for colorectal cancer, other one for lung cancer, kidney cancer, gastric cancer, breast cancer, glioblastoma. That's brain cancer. Not only that, there are other diseases that are very serious for patients not only cancer, but it turns out that one of the leading, probably the leading cause of blindness in the United States is a disease called macular degeneration. This is something your grandparents may have. And what happens there is, again, abnormal vascular proliferation in the back of the eye, leading to hemorrhage, leading to blindness. There has been no medical treatment, no drug treatment for it up till now. The only treatment was lasers, photocoagulation. But now, for the first time, there are drugs, first Macugen, and now Lucentis, which can actually reverse macular degeneration. So about 10 million patients a year are now using these drugs. But more broadly, you can not only use this kind of approach for helping the angiogenesis field, but you could use it for any drug that is sort of hard to give to patients. Here, just as a proof of principle, Larry Brown, one of our graduate students, delivered insulin in a tiny little pellet in diabetic rats for 100 days. And, but really, we did all this, and then the question is, how do you get to the point where it's going to really help patients? And again, 28 years, it was a long time. So one of the things I got involved in, I'm going to talk about this next in Jack Gill's class, is that we actually got to get involved in licensing our patents on this and starting companies. And as a consequence of that, many different products based on these microspheres emerged. And here you see different ones, like, for example, if somebody has advanced prostate cancer, now there's a drug that people would give, luteinizing hormone, releasing hormone, but the problem is you can't swallow it, you can't put it in a patch, it's just too big. If you inject it, it's destroyed right away. So they put it in a microsphere. Originally it lasts for a month, now 4 months. Just as a second example, how many people here saw the movie A Beautiful Mind with Russell Crowe? So do people know what disease did he have? Does people remember? Schizophrenia. So he saw people who weren't there. Does anybody remember how he was treated medically in that movie? He got shock treatment, but electric shock treatment. Today, however, he'd probably get this, Risperdal Consta. This is actually based on the microspheres. It's a drug that has about $1.5 billion of sales a year. And there's a very good drug. You give it an injection once every few weeks, and it's had an enormous effect on preventing hospitalizations and suicides and things like that. But you always want to do better. And one of the ways we, actually, I think I'll just skip a couple things. One of the ways we started thinking about doing better was, could we come up with better materials? And one of the things that I started looking at was, how did materials find their way into medicine? How do materials, so this was actually very interesting. As I mentioned, I worked in a hospital, and I was a young engineer. And as I started thinking about how materials found their way into medicine, I thought I'd do an analysis of how this happened. And what I found was actually very surprising. I found that almost all the time when materials found their way into medicine, it was not driven by a chemist or a chemical engineer or material scientist. It was almost always driven by a medical doctor. And what they did, because medical doctors urgently want to solve medical problems, is they'd go to their house and they see what kind of object kind of resembled the organ or tissue they were trying to fix. And then they'd use it on a patient. Let me give you an example, or actually 4 examples. So these are all true. People may sometimes think these are funny, but these are actually true. So in 1967, in the case of the artificial heart, some of the surgeons wanted to make an artificial heart, and they said, what object has a good flex life like a heart? And they said, a lady's girdle. This is actually true, though. And then it's made out of polyurethane. So they decided they'd make the artificial heart out of the very same thing. That was 1967. Now we're in 2010. That's still what it's made out of. Because once you start down that path from like a regulatory FDA standpoint, it's very hard to change the material. Well, why has the artificial heart not worked very well? Well, the biggest reason has been when blood hits the surface of the artificial heart, the lady's girdle material, it forms a clot. That clot goes to the patient's brain, they get a stroke, and they die. But if you think about it, something that's designed to be a lady's girdle is probably not the optimal blood contacting material. And this problem pervades all of medicine. Dialysis tubing with sausage casing. Vascular graft, that's an artificial blood vessel. That was a surgeon in Texas going to a clothes store to see what he could sew well with. Breast implants. One was a lubricant. The other actually a mattress stuffing. You can probably think of the logic. So one of the things that you learn in engineering is design. So we started thinking, maybe we could do better. Maybe rather than take these things off the shelf, what if we ask the question, what do we really want in a biomedical material from an engineering standpoint, from a chemistry standpoint, from a biology standpoint, and then could you synthesize it from first principles? So we started with an example. And the example that we started with was that when we started, there was only one family of polymers that were FDA approved that were synthetic and degradable and implantable. These were polyester sutures. And they dissolve like this. They start looking like this. Then they get spongy. Then they fall apart. And this might be okay for delivering some drugs, but if you had a really toxic drug like insulin or an anti-cancer drug, if it dumped it out, it could be incredibly dangerous. It could kill you. So from an engineering standpoint, you'd say, well, what do you want? And what you'd say is that you don't want the polymer to dissolve like this. You want it to dissolve like this, surface erosion, like the way a bar of soap dissolves or an onion peels. How do you do that? Well, the way you do that is, again, you go through a pretty detailed engineering design, and I won't go through all of it, but just briefly, if you were going to start from scratch and make a polymer to do this, you could build in whatever you want. So you could ask different questions. First question might be, what should cause the polymer to dissolve? Should it be enzymes? Should it be water? Should it be something else? Well, enzymes, we all have different enzymes. We all might have different enzyme levels in our body, so that might not be so good. But all of us have excess water. So we said, if we want to make this reproducible from patient to patient, let's make water the catalyst. Then we go to the next question. Say, if we want to get surface erosion, well, then we need to make the polymer we thought repel water, be what we call very hydrophobic, to keep the water out. Then we'd say, but how will we still make it degrade if we do that? So we said, well, the bonds, the chemical bonds that connect these hydrophobic units should be very water labile. Very water labile. And so we actually calculated by going through organic chemistry that it had to be the anhydride bond. So we actually found a very good use for organic chemistry. And we proposed that, and then we even went, we got very good chemists and toxicologists like Mike Marletta, who was used to be head of Berkeley's chemistry department, and we worked out this kind of polymer. And this polymer is very hydrophobic, has 2 units, and it's connected by water labile and hydride bonds. But what's exciting is not only does it show this type of erosion, but if you change the ratio of this to this, we can make the polymer dissolve at almost any rate we want, from 0% of the 1 to 79%. So you see, this will all be gone in 2 weeks. This will take 3 or 4 years. So you could just dial in the ratios of these and make them last almost any length of time. Now, one of the things that I've mentioned to you is that I really wanted to do when we did this work is to have a medical impact. Could we help people? And in 1984, Henry Brehm, who was a young neurosurgeon, just starting at Johns Hopkins, wanted to have a better way to treat brain cancer. And let me just tell you a little bit about that. The worst form of it and the most common form is glioblastoma multiforme. Regardless of how you treat the patient, the mean lifespan is less than a year. The drug in the 1980s that was most commonly given, and probably for 20 years most commonly given, was this one for brain cancer, BCNU. But it was terribly toxic. They gave it intravenously. It would travel and cause terrible side effects on the liver, kidney, and spleen. So we thought we could introduce a new idea. Local chemotherapy using the polymers. Could we allow, and this is just theoretical at this point, the neurosurgeon to operate on the patient, which he's going to do anyhow, to remove as much of the tumor as they could, but before they close up the patient, line the surgical cavity with a polymer that slowly releases the BCNU. Now, normally this has got a 12-minute lifetime, but if it's put in the polymer, it would be protected. It would last as long as the polymer lasts. So what the neurosurgeons wanted was a polymer that was degradable, so it wouldn't accumulate in the brain, that had the surface degrading properties, so it couldn't dump the drug. And from animal studies they wanted it to last for about a month, and we could do that by changing those monomer concentrations. And most importantly, what we're doing is what I'll call local chemotherapy. High concentrations in the brain where you want it to be, and low concentrations in the rest of the body where it might cause harm. Now, the thing is, there's a number of professors here, and they, like myself, whenever they have an idea like this, like a new polymer, new application, you have to raise money. And the general way you raise money is you write grants to the National Institutes of Health, if it's health-related, which this was. And there's study sections, which are composed of professors at other universities, and they decide how well you do. This didn't do very well. I actually saved the reviews. I titled this slide, "This Approach Will Not Work Because." In 1981, when we first proposed this, the reviewers, the chemists said, well, you'll never be able to synthesize these polymers. But I had a very good graduate student named Howie Rosen. Howie later would become president of the Alza Corporation, very successful drug delivery company that Johnson & Johnson bought for $12 billion. Howie also was elected to the National Academy of Engineering a few years ago as well, and he synthesized them. But then the reviewers said, well, the polymers are reactive, they have anhydrides, they'll react with whatever drug you put in. I had another couple postdocs. Bob Linhart, who's now Constellation Professor of Chemistry at RPI, and Cam Leung, who's the James Duke Professor of Bioengineering at Duke, and they synthesized these and worked out ways to not have it react with drug. But then the reviewers came back again. By the way, at NIH, as the professors know, they're not very fast. So the reviewers said, well, they still shouldn't fund it because the polymers are fragile. They're low molecular weight. They'll break in the body. But I had another couple postdocs, Edith Mathiowicz, who's now full professor of bioengineering at Brown, and Avi Dome, who later would become chairman of medicinal chemistry at Hebrew University. And using the right catalysts and other things, they made them very strong, very high molecular weight. And then the reviewers said, well, they still shouldn't fund it because nobody's ever used for 20 or 30 years a new material in a person. It's got to be toxic. I had another graduate student, Cato Lorenzen. Cato's now Dean of Medicine at University of Connecticut, member of the Institute of Medicine of the National Academy of Sciences. And he showed, which actually proved some of what Mike Marletta told us, that they were actually quite safe. Anyhow, this kept going on until 1996, when the FDA approved this treatment. It was actually the first time in over 20 years they approved a new treatment for brain cancer, first time they ever approved this idea of polymer local chemotherapy. You can probably tell from the way I'm speaking that I'm very proud of how well our graduate students and postdocs did. They became chairmen or presidents of major corporations, held endowed chairs, chair people of major departments. The reviewers, on the other hand, they haven't done so well. Now, I'd like to actually show you what this operation looks like, but if anybody's squeamish and doesn't like the sight of blood, you probably shouldn't look. But let me just show you. This is going to be human brains. They put a disc in. They're about the size of a dime. Usually they put 7 or 8 in and close the brain up. By the way, it's very hard, to get good advice when you give a talk, but about 10 years ago, I was giving a lecture at MIT, and my wife came to the talk. And at the end of it, I said to her, Laura, I said, what did you think of the talk? She said, well, Bob, she said, the talk was all right. That, by the way, is extremely high praise. But she said, I don't know if you were watching, those engineers, but she said, you had those 2 slides on for 10 minutes, and all those poor engineers were turning green and looking at the floor. So ever after that, what I did is I always did what I did today. I gave people a warning and showed them very quickly. But I want to tell you a sequel to this, which was that 2 years after that, I actually got asked to give a dinner speech. But this was actually to a group of surgeons and neurosurgeons. And I told them the same story. And at the end of it, a bunch of the neurosurgeons came up to me and they said, those 2 bloody slides that you showed? And I said, yes. They said, those were fine. They said, no problem. You could have left those on as long as you wanted. They said, but those chemical formulas, to leave those on for a neurosurgeon, I guess is terrible. Hopefully there's no neurosurgeons here. But at any rate, let me just show you a little bit of the clinical data. This is at the end of a year. You have 61% in the treated group, 19% in the control group, which are the best common treatments. At the end of 2 years, you get 31% in the treated, 6% in the controls. Now, there's various things that you can sort of feel proud of when you do things. One might be, coming up with a good idea, and other people could say, when Carrie gave me this nice introduction, getting patents or awards. But what was really interesting one night, and I didn't realize this until my sister and about 25 people in the lab called me up, is I said, Bob, did you know this was actually on television? Now, that's really an accolade that you never expect. And there's a TV show, which I didn't watch, called ER. How many of you ever watched the TV show ER? Anybody? A number of people? Well, let me just give you a little background on this, and hopefully this video clip will work. So this is actually from many years ago, maybe about 10 years ago, on ER. So ER, for many years, the hero of it was a guy, a clinician named Mark Green. And Anthony Edwards was his name in real life. And it turns out that all of a sudden he gets a brain cancer. And he goes from person to person doctor to doctor, and they all tell him he's going to die. Finally, he goes to one last doctor. And I'm going to just show you a clip. By the way, the doctor who plays this is actually Susan Sarandon's ex-husband, Chris Sarandon. He's actually quite arrogant, as you'll see. By the way, and I don't know, just in case people don't know who Anthony Edwards is, did anybody here see the movie Top Gun with Tom Cruise? So Anthony Edwards, that was Tom Cruise's partner in Top Gun, he actually didn't do too well there either. He died in that too. Anyhow, let me show you the video. Let me show you the video. And what I want you to know, the major thing, and I'll try to highlight it if I can remember, is they're going to talk about a chemotherapy wafer. That's ER speak for what I'm just telling you. So let's see if this works. Here's Anthony Edwards. Can we make sure this is loud enough? I don't know. How do I? Where was the guy who? It worked before, but yeah. Yeah, it's just it was on. Yeah, I know. Well, we need one that. Where was the guy that was here earlier? He seemed to know what he was doing. Yeah, that's definitely it. But he had it before because I checked it. I'm just not sure. It's actually a very good video, but because he had it very loud. Yeah. Because it was actually pretty loud when I came in. But what did he do? Well, if we can't do it, I don't want to hold it up. It's just pretty. Oh. Is it ready? Oh, you got it. Here, we could probably. Yeah. Okay. Let's see. Can people hear it?
Rachel called. She wants to come out today early for Christmas.
Agreed.
That's the doctor. Now it's.
Jimmy, can you just mask the bottom?
Oh, this is it. It's okay. We can keep it here.
Come over here. Let's see a lesion like yours. Lesion is inoperable.
Is there some? If it doesn't work, we'll stop it. I'm the last person to do it. Okay, so what do I do?
Just go back on the slide.
Go back and then forward? Okay. Needs to be louder. Needs to be louder. Doesn't. Well, it's okay. I'll tell you what happened. I won't do nearly as good a job acting. Yeah, it's okay. I think it's.
what he's doing.
Yeah, well, he's showing in the brain images of the brain cancer right there. And then what he's doing is he's going to tell him how he's going to treat him. Basically, he's saying he went to all these other doctors. They said he was going to die in 2 weeks. And now what he's doing is he's going to give him a new treatment. The new treatment's actually the chemotherapy wafer that I just showed you. And he said, you know, and so then it sort of keeps going. And at the end, you know, you see they're actually very happy. They're going to hold hands. And then he's going to say to them, you know, as they're holding hands, by the way, this is at the end of the year. It's like December 31st or something like that. So what he's going to show them as he's explaining it and how he's going to be all better, hopefully for a long time, is that even though it's going to be a new year, let me just see, I just want to let it go a little further. Yeah, he's getting happier now. And now he's getting a lot happier. And let's see. Yeah, you can see he's doing much better. This guy's telling him, and his wife's very happy. They're holding hands, like I said. And what he says, the neurosurgeon says to him, he says, well, now you know you'll be able to enter the new year with a load off your mind. He said, that's brain surgeon humor, meaning that he's taking out the tumor and putting in the chemotherapy wafer. Anyhow, I'm sorry that that didn't work out so well. And by the way, he did not live forever. He lived for many years longer than he normally would have lived. What happens is that now actually what they're doing is that there's one other treatment, Timidar, and what you see here is now that's a new drug, and you combine that with the wafer, and that probably, again, extends life longer than ever before. The broader point, though, that I wanted to make is that one can use this idea of local chemotherapy not only for cancer, and that's had an impact, and they're using it for different kinds of therapy, but where it's had even a greater impact is in the whole area of drug-eluting stents. And just to go over that, it's really the same idea. If somebody has a heart disease, they put these stents in. They're like a Chinese finger puzzle, but about 50% of the time, within 6 months, they close off. And now what they're doing is exactly what we developed. They're taking polymers, they're putting like anti-cancer drugs like Taxol in them, and that's had a remarkable effect on preventing what's called restenosis, which is this problem. So you can use this principle in a number of cases. Let me show you one other example. Whatever you think. Okay? I should leave it up to the audience. You want me to go back? Even though I did such a good job explaining it. Okay, so do you want to try it? I'll let you do it, since yeah, now you hear it. You want to go back one and then put it on? Can you turn it up a little?
Rachel called. She wants to come out.
very, very modest. Thank you very much. Okay, I'm going to skip forward a little bit. And the area that I want to turn to end the talk is the idea of combining materials and cells to create new tissues. And this is like a really tragic problem. There's many situations where there just are no drugs. And so here's a little baby who's actually dying of liver failure. And the only way a child like that lives is somebody else dies. And here I've worked very closely with a surgeon named Jay Vacanti, who's one of my good friends. He heads the pediatric surgery at Boston at Mass General. And to solve this problem, or to address this problem, what he and I proposed, this is from an article in Science. We actually originally wrote this down in 1983. But we proposed this idea where you could take isolated dissociated cells. And our thinking was if you put them into the body at random, not much happens. But cells are intelligent. So if you take these same cells and put them close enough together, and our thought was to put them on a polymer scaffold, they could actually communicate and create a tissue. And if you figure out the right way to grow them, and this is actually a big engineering issue, that maybe you could make the tissue. So you have to figure out the right nutrients, the right scaffold, the right bioreactor. Just as a quick example, when we tried to make a blood vessel, it's very hard to make a blood vessel. But the way we were able to make a blood vessel, as we published in Science, is we actually hooked up the scaffold to a pulsatile pump, your heart, and then it worked. At any rate, and then you could make these different tissues. So I thought I would just give you a few quick examples of tissues we tried to make. And the first one is, I'm just going to show you a scaffold with cells on it. But let me also show you, and I'll pick cartilage as an example, what I expect might happen 30 or 40 years from now. And if somebody comes in 30 or 40 years from now, let's say they would like a new nose from a plastic surgery standpoint. And Prasad Shastri, one of my postdocs, he now runs an institute in Germany, actually worked out a way using CAD/CAM techniques, computer-aided design, to make things in any shape and make them 98% porous. And here he's done a nose. Now this is kind of like a regular nose, but let's say somebody you could have a computer screen predict any nose you want. So let's say somebody wanted an upturned nose. It wouldn't be so hard to do. You take a little bit of this off. Let's say somebody wanted a hook nose. I mean, they probably wouldn't. But if they did, you give them a little bit more stuff. At any rate, Chuck Vacanti, one of our collaborators, has actually done this on nude mice to illustrate the principle. And here he's redone this guy's skull, this guy's cheek. If you open the animals up and look at it, it's pure white glistening cartilage. Now, one of the things, and here's an example of what's called histology, where you can actually characterize it and see that it really has all this is almost like out of a textbook of the different things in cartilage, glycosaminoglycans, the right kind of collagen, the right kind of cells. Now, one of the things we're working on with the Army is they see patients who come back, or soldiers who come back that sometimes don't have ears or other problems. So we've actually been working on that. Linda Griffith, who worked with us, made a scaffold in the form of a human ear. And here's a high-powered SEM, scanning electron microepigraph of it, with cells on. And over time, the cells are going to make matrix, and you'll get all cells in matrix. Now, Chuck actually didn't put this on a human yet, but they have put it on rabbits. And here's the rabbit with the human ear. He hears fine. And this has been tested now on patients. Jay, my collaborator, here's actually a 12-year-old boy. But when you look at this 12-year-old boy, he's got no chest covering his heart. But like other 12-year-olds, he likes to play baseball. But you could imagine if he ever got hit in the chest with a baseball, he could die. So Jay operated on him. We took his cells, made a scaffold, and made him a new chest. And he's doing fine now. Let me give you a second example. This example is based on a license to a company we did. And here's a little boy who's very badly burned. But you could take a product based on this work, which is neonatal skin fibroblasts, put it on this polymer sheet, cryopreserve it, and if you put it on the patient, it looks like this. This is at time 0. But let's come back 3 weeks later. Let's come back 6 months later. And he's pretty much healed. So these are now approved for patients who are burned or patients who have diabetic skin ulcers. Very last example to end this talk is, what about people who have spinal cord injuries? Could you ever help that? So here, Erin Levick, one of our students, she's now a professor at Case Western, worked with Ted Tang and Evan Snyder, who's now at the Burnham. And here we actually made a scaffold that mimicked the gray matter and white matter of the spinal cord. And we took Evan's neuronal stem cells and put them in this. And let me just show you the pictures. This is the scaffold. This is the inner part that's very porous. The outer part, which she's created in an oriented way to help axonal guidance. And what we did in these experiments is, just as a proof of principle, is to make the rats paraplegic. And the way we made them paraplegic, which means they won't be able to move their hind legs very well, as I'll show you, is we took out a certain portion of the spinal cord in this hemisection. And we did 50 animals, followed them for over 400 days. All these, by the way, were done within 24 hours after the injury. But let me first show you the rats that were done. Fortunately, these rats don't talk, so we won't have the same problem. But we'll watch the rats, and this is a typical control. There were about 38 of these, and this is the mean of them. Could be cells by themselves or polymer by itself. But the two things to notice are that they're not able to really bear his own weight, and the other thing to notice are the paws in the back are splayed in a very awkward fashion. We'll just watch this for a few seconds more. OK, there he goes. Let me now show you what happens if you put the implant in. And this, by the way, is 100 days after the start of the experiment. If we put the implant in, let me just show you what happens also at exactly the same time. It's not a cure, but what you'll see is this guy, even though he's a bit heavy, is able to bear his own weight in contrast to the other. He walks a bit clumsy, but he's doing much better. There's actually a quantitative scoring system, BBB system. He gets about a 14, whereas the other gets a 5. 20 would be perfect. But he bears his own weight. Notice the paws splayed in a much more normal fashion. Those are part of the scoring system. My wife tells me not to leave that on either for long. But just to conclude this, so that's where we were up till about a year ago. But we, again, want to move this to patients. And the next step in moving this to patients is to do primates, monkeys. And so the next work I'm going to show you is preliminary. We've only done about 6 monkeys, but it's actually very exciting. We've done the exact same experiments. These are done by really top spine surgeons, Eric Woodard and others. And basically, we've taken primates, and I'll just show you what happens with them. Again, we're taking out a portion of the spinal cord on one of the legs, which is going to make him have a very hard time moving one of his legs. And just to show you that, let me just show you one set of monkeys. So this is pretty typical of the control. This is at about 8 weeks out. See how it's having trouble moving this leg on a treadmill? And in a second, hopefully, you'll see the treated animal. And notice he's doing much better, just with the spinal cell implant. It's a neuronal stem cell. And we've actually done this on several sets of monkeys. So I'm going to show you on the final panel, the second set, if I can get this. I'm doing it, but well, it's going to be pretty similar, but maybe we'll stop. If you know how to do it, I'm not sure what I'm doing. Is that the line? Yeah, that's the one we just saw. Right? Yeah, so yeah, I think it's going to be similar, but yeah. So if we go to the last, no, maybe that was the other one. That was the last. Yeah. But basically, it's the same kind of thing. It's just at a later time point. So basically, the point is whether you do the rats or the monkeys, they're promising. A long ways to go. And obviously, animals aren't people, and we're still doing it 24 hours after the injury. But this is a combination of a spinal polymer implant with the stem cells. So let me stop here. It's really been an honor to be here today. I apologize for the videos, but I hope people get a flavor for how one might use bioengineering to try to address important problems in angiogenesis research, drug delivery, and ultimately someday tissue engineering. Thank you very much. Happy to answer questions. Sure.
Let us take a couple of questions.
Yes.
One of the themes you touched on a couple times during the talk was this idea of having to shepherd good ideas through kind of a very long process. How do you do that? So you had one example where you had a biomaterial it seemed like it never got approved by NIH. You still managed somehow to advance that.
how do we do? Well, I guess ultimately the key is raising some money somehow. And so originally, I'm not sure if that was what was behind the question, but basically, so for about 5 years, I kind of bootlegged it on other grants. And then, even though I could never get the government to fund it, we were able to get a company to license it and give us a grant. And that company gave us several $1 million, and that really helped us. And though it's interesting that after it got approved, then the government started giving out very large grants for other drugs you could put in these polymers, which is sort of typical. You know, you have to, so it took us many years, you're right, and that's often what we've had to do. I think we're not alone in that. I think generally what people find, unfortunately, when you're competing for grants is that there's a tremendous amount of conservatism. And so you probably, if you have a good idea, have to bootleg it somehow or find some non-conventional source for funding. But that's how we did it, and we just kept at it for a long time. Anything in medicine, sadly, takes a long time.
Well, rather than have more questions, okay, let's have one last question because we're running late. Apologize for that, please.
So for the spinal cord injury, I assume you just have a limited window between when the inflammation abates and scar tissue sets in, and then you away from.
I think that's a great question. She's saying, what's the window in terms of time? And I think I expect what you said is correct, but we don't really know. I mean, we haven't really done detailed studies to find out how long or short that window is. But I would expect that that's right, though I also expect that, and we've been looking at this too, which I didn't say, is that you could probably also help this not only by cell therapies, but by, and we've been doing these in other studies, is by releasing things like steroids, for example, to reduce the inflammation, to release neurotrophic factors may help. So we actually have separate experiments going on in all these areas. And it may be, again, there's plenty of people looking at this, that someday maybe it's going to be combining these things that could make that window as broad as possible. Thank you.
Well, I want to thank the Store family for endowing again this wonderful lectureship series, Dean Curtis for his support, and Angie Louis for organizing this. Professor Langer will now go to the business school for another lecture, and then we're going to have him again tomorrow morning at 10 o'clock. He's really giving us a lot of lectures for his invitation, too many lectures perhaps, but tomorrow morning at 10 o'clock we're going to have him in the genome building.